A pleasure to be here, and thanks for coming. So the program is called Non-Growing Microbes, and it may seem to you, if you come from white dwarfs or from multiscale flow, it may seem to you very specific, but I'm going to argue that actually we study non-growing microbes, but actually we are using non-growing microbes to ask very, very general questions about what is life, actually. So what is life? I think it's quite accepted that life can be defined at the level of a cell, right? If we understand the cell, more or less we understand life. Of course, there are things that are more complex, like development and how higher organism, but the mystery of life is already fully there at the level of a single cell, okay? And a single cell can be one of our cells, but it can be also a microbe. And actually, if you look at life, microbe may be called simple life, but there is no simple life. Immediately as you talk about a living organism, a living cell, you're talking about of the order of magnitude of 100,000, 10 to 100,000 of different molecular components, right? So it's not the number of molecules, it's different degrees of freedom, different molecular components in the cell. So there is no simple life. Viruses are not, from the point of view of what I'm talking about, are not considered life, right? Live. So how do you describe a system where actually you have no simple system to describe it? You have immediately to dive down and address the whole complexity of life. Okay, there is no simplification that can be made, at least by trying to look at it. Now, how do we picture a microbe or a cell in general from a simple picture that can be understood, I think, by physicists is that actually all these components can be seen as some continuous axis. For example, you have one of these components, let's call it Xi. It has some concentration in the cell, right? So Xi would be component I will be represented by the concentration of this component in the cell. For example, it can be ATP, which is a very important molecule. How many ATP there are in the cell, and then it will be represented by one axis in this dimensional space. And in a cell in general would be represented in this 100. I don't know how to draw 100,000 dimensional space, but this is what a cell is. It's one point, one vector, in this 100,000 dimensional space. This is what a cell is at a certain point, time t. So you can label these vectors, and these dimensions, each dimension, as I said, is describing the concentration of a molecular component in the cell. And the state of the cell at time t is this vector, which actually is this. All these numbers and are functions of time, okay? So that when you look at the cell, in principle, over time, it would be a trajectory in this space, okay? And as I said, please do interrupt me if anything is unclear or if you have a comment. So in principle, you know, and there are some approaches like that. Yes?
So what about all these structures inside cells? They're not just captured by a single number like concentration.
Okay, thanks. Thanks for the question. So for the sake of this talk, I'm going to completely ignore spatial inhomogeneity, and I'm going to assume, which is of course an assumption that needs to be verified, that because we are talking about microbes with a dimension of about 1 micron cubed, I'm going to ignore spatial inhomogeneity. Okay, well-steered environment. Okay, so this is a picture, and as you can imagine, you could, in principle, start to try writing equations for the time evolution of these trajectories, right? And it's going to be a very complex function that are going to depend on many parameters. And I think what physicists have been doing since the days of system biology and quantitative biology is to try to reduce the dimensionality of this problem, right? And one way to reduce the dimensionality of this problem is, for example, just to focus on some particular property of the cell that actually, for which actually you can ignore most of the axes and concentrate, let's say, on 2 or 3 axes, 2 or 3 components in the cell, and try to write a few equations, measure very precisely the experimental parameter, and establish a small model for this, which can be predictive, and there are very nice examples of such an approach. Yes?
Is there a fundamental reason that you have to start at 100,000? The complexity of the dimensionality of life starts at 100,000 elements. Why not 10? Can you finally make a living organism? It would be a lot simpler.
It would be a lot simpler for us. I think it's an open question, right? My intuition is that life needs all these dimensions to evolve, and maybe the origin of life has not started just by one molecule replicating and another. It started from conditions where hundreds of thousands of molecules found themselves in the same place, maybe, but it's really very speculative. So what we're trying to do is to describe these dynamics. Now, when we say, non-growing microbes, right, what do we mean? So what do we mean in this picture of a cell when microbes are growing or when cells are growing? Typically what is going to happen when a cell is growing is that actually a cell grows, it increases its volume, and then it divides into 2 cells, which are actually the initial conditions for growing again and dividing again. So it's a cyclic, we call it the cell cycle because it's really a cyclic behavior. And for example, for our cells, you can really see, if you look at some of these compounds, you can really see oscillations, and each time there is a peak, there is a division, right? So it's really a cyclic process. And typically in this picture, a cycling cell, a dividing cell, would be some limit cycle in this space, and where the trajectory goes over and over in a cyclic manner. So when we mean a non-growing microbe, which is the topic that we are studying, we mean that actually from this cycle, the cell actually goes into some other dimension of the space where this process of growing is arrested. Now, why are we interested in that? We're interested in that because in the microbiology field, 99% of what has been done is on this limit cycle, okay? Whereas if you ask ecologists, 90%, 80% of the bacteria in the world are actually in the non-growing state, okay? So one of the questions that we asked in our program was first, okay, we know per definition that we are not in this limit cycle of growth and division. What is this non-growing state? Because it has been not that much studied, is it one state? Is it a state at all? How do we represent it in this high-dimensional space? And how do we characterize these non-growing states? So one of the nicest examples of these non-growing microbes is what is called spores. And this is maybe the extreme example of what life can be in the non-growing state. So spores are bacteria, that emerge from these cycling bacteria. When these cycling bacteria encounter conditions that are stressful, for example, no more food around, right? And food would mean a mass, right, for constructing a new microbe, and energy. This is typically what we need. So when there is no mass and no energy around, these microbes will actually leave this limit cycle, This limit cycle for this microbe in rich condition typically takes 30 minutes and go to a state which is quite far away from this state in this dimensional space, to a state which is really, and in our view, and I have to say that all this picture of limit cycles and attractors is still open and controversial, but this is what we think when we think about states of cells. So these spores are actually far away from this, and despite the fact that the cycle itself takes 30 minutes, it takes about 8 hours to go from here to here when the mass and the energy are depleted. But once they are here, and why it takes 8 hours? Because they're going to completely change their structure. They're going to actually crystallize the middle of the cell. Diffusion inside these spores is prevented. You can really see it as a crystal whose main goal is to keep all the components against entropy, against degradation, against all the things that are typically encountered by the bacteria that are growing. And then these spores, once they are formed after these 8 hours, they can survive for at least thousands of years without any supply of energy or food, right? So, and they are alive because as soon as you supply them with energy, so when you restore energy and food, they will go back to this limit cycle within 10 minutes. This is typically the time scale. Now, why do I argue that this is an attractor in this space? Because actually you can do many perturbations to these spores. You can heat them to 70 degrees. You can put acids, antibiotics, phages, viruses of bacteria. They're going to survive all that. And if we say that it's an attractor state, which is also interesting, is that you can take bacteria from this limit cycle. You can take bacteria and really perturb this limit cycle with all kinds of things. They will all, at the end, converge to this point. And if you look also at the distribution of times that it takes, so you have, instead of one cell, now you take 1 million cells, right? This is what is nice about microbiology, is that you can work with large ensembles. So you take 1 million cells, put them in this state. You can keep them in this state for a month, and this is an experiment actually that we did. And when you put them back into mass and energy, and you plot the distribution of revival times, so namely the time it takes for each bacterium to go back to growing. Okay, and we can look at it we can look at them under a microscope and really pinpoint this time. So for each cell, we look at thousands of cells, and for each cell, we mark the time from being the spore to being a growing cell. And the mean, as I told you, is around 10 minutes, and the distribution is also around 10 minutes. So it's very narrow. And if you now, instead of keeping them for a month, you keep them for a day or for 2 months, the distribution is exactly the same. So, okay? So there is no deterioration. This state is really an attractive state. And this is, any question? So this is the extreme example. Okay, so and actually this is a non-growing state that has been relatively well characterized, at least in some bacteria. But it turns out that most of the bacteria do not make these spores. Okay, and if we look at the bacterium that is the hydrogen atom of microbiology, namely E. coli, I guess you've all heard about E. coli. So E. coli doesn't make spores, right? So what happens to E. coli? It has also a limit cycle when it grows of about 30 minutes. What happens when you deplete mass or energy, etc.? Where will it go? So if you deplete mass and energy for a long period of time, certainly for a month, and even much before, it will die, okay? So none of the ability of spores. So it means that in this space, so it will, you deplete energy and mass, which allows this dynamical state, right? So it will now make a different trajectory in space, reach somewhere else, and eventually die, which means that in this high-dimensional space, death is there all the time, and bacteria and the cells may reach this irreversible state from many different locations in this high-dimensional space. But what we see is that actually E. coli is adapted, at least for shorter period of time, 1 day, 2 days, 3 days. It is adapted to go into a state which is maybe closer to the state for which it doesn't need 8 hours, but much less time, and in which it can survive for a few days without mass and energy supplies, and stay there for a certain period of time. And these are also the states that we were interested in our program to map this trajectory. It's unknown, right? So one has to realize in biology, you know, I grew up as a condensed matter physicist until the end of my PhD, and I worked on gallium arsenide, and we had this big book on gallium arsenide telling you the sound velocity, all the electric constants, everything, right? It's a big book with all these constants. What needs to be realized is that in biology, we are 200 years behind, right? So everything that you touch, there are many unknowns, right? And even that, I'm telling you, the bacterium that has been the most studied for the past 100 years, we are asking, when we starve it, does it have 1 state, 2 states? Does it have state at all? Are there attractors there? Are these trajectories? We don't really know, okay? So what is clear is that in order to maintain life, not in this crystalline extreme spores, what I think is an emerging theme of the program is that you need a supply of energy. So if you completely take out the supply of energy, you are going to reach death much faster. And there was an amazing example here by Carrie Hartwood of a bacterium where you can actually disentangle between mass and energy, because she studied bacteria that receive mass from a sugar. Okay, so the sugar supplies atoms, right? And receive mass from also from CO2. But can receive energy from light. So it's a bacterium that can do photosynthesis, right? And what she showed is that if she just, a very simple experiment, she just give them mass and energy at the beginning, they multiply exponentially, and then they do so on a log scale versus time, the number of bacteria will look like that, right? So they grow exponentially, and eventually they deplete all the initial mass that was given, so they stop growing, and you can see that. And what she saw is that if she doesn't give them light, which is only an energy source, they would die. But if she provides them with light, they can live for months. Okay, so that's a beautiful example showing how just the difference of providing energy or not providing energy is making all the difference. And for E. coli, from several of the talks that we heard and from the groups that were in our program studying the membrane potential, for example, you need, in order to maintain life, you probably have to maintain the membrane integrity and the membrane potential that actually isolate the bacteria from its surrounding. It's very important. There is some maintenance energy, and there are people here that measure this maintenance energy required to maintain life for a longer period of time, and if you don't supply this maintenance energy, bacteria will die. So it means that whatever the state is of non-growth, this state consumes energy just to stay alive. So this is something that we are studying. And the question that one of the questions also that we asked is what happens when we perturb the state? So we don't let it nicely deplete its new mass and energy, but we actually do something more severe to these cells so that we don't allow them to continue on this limit cycle. So, and this is where, the title of statistical physics comes into our thinking. Because what happens in all what I've described to you, for example, the sporulation process, this process that takes 8 hours, is a very precise and beautiful trajectory in this space on specific axes that were mapped. And we know axes, for example, are genes. These genes goes up, these genes go down, this goes there, this phosphorylates a protein, never mind. So it's it's what we call a regulated state, from the term gene regulation, a regulated state, and it is a very particular trajectory in this phase space. And when you look at it, it's not clear what, as a statistical physicist, we can say about this trajectory. I'm sure there is something to say, but it's difficult. And actually, a review by Giorgio Parisi about 30 years ago, which is called Statistical Physics and Biology, and I quote him, he said, even if we succeed, and this is related to this high-dimensional space with n variables, if we succeed in modeling a unicellular organism with a system of differential equations, this is that, with n variable, we would still have to deduce the overall characteristic of the behavior of the system. It is the same problem as in statistical mechanics, where knowledge of the laws of motion does not directly imply an understanding of the collective behavior. And going along these, he postulated that the study of disordered system will become the conceptual framework for understanding the dynamics of living organisms. And this was 30 years ago. Now, what we claim inspired by this approach is that maybe in the regulated state, trying to have these laws for characterizing the behavior of the system from a higher level is really difficult. But what happens if we take this cell here and we perturb it so strongly that now it's doing more or less a random walk in this high-dimensional space? What can we say then? Can we ignore all the details of the dynamics, the precise parameters of this differential equation, and say something more meaningful? This was a question. And the idea of how maybe to start addressing the problem came from an experiment that we did, where we took cells in this state, in this limit cycle, and very abruptly we expose them, so they are in this limit cycle state, they grow nicely, and then very abruptly we expose them to a stress of a certain duration, t weight, and then we take away the stress, put them back exactly in the same initial condition, and look how the system relaxes to the state it was, namely how long it takes for it to go back to this limit cycle. And when we do that, so initially at this state, then it's going to be very perturbed, so everything goes down, and then eventually we take it off, and then it relaxes back to its initial value. And what was interesting, and it was a very simple experiment to do, what was interesting is that when we varied the duration of the stress duration, what we saw is, first of all, the relaxation back to the initial state was shifted to longer and longer duration, which is not very surprising, and also the time span over which this relaxation occurred was becoming wider and wider, and the relaxation back to the initial condition was very non-exponential. Okay? So three features: non-exponential, relaxation, dependence on TW, right? Okay, and strong dependence of TW. We'll start with these two features. And now, what it reminded us is, and this is a demonstration I'm carrying around because it's really fun to do, it reminded us of the theory of aging in physical systems. So if you take a physical system with many degrees of freedom, for example, this Mylar sheet here, when you apply, and these are experiments that were done in physics, right? So if you apply a weight on this Mylar sheet, right, for a time TW, so this is the perturbation that takes for TW, and now you leave the weight for this time, and then you relax it, and you measure the dynamics of relaxation back, right? I don't know if you see it. You have the very fast time scale, and you have, but even after the fast time scale, you can still see, I didn't touch it, you can still see relaxation occurring on many different time scales, okay? And even if you come back a week later and you are careful, you will still see some changes, right? So this is this very slow relaxation, and the graph describing this behavior are very similar to our graph. So the longer you put the weight on these Mylar sheets, the longer the relaxation back. And I guess many of you have already understand that it's also it's described very nicely by statistical physics with a model of spin glass. So aging, for example, in the Sherrington-Kirkpatrick model can be described simply by having a network of interacting nodes, and these interactions can lead, when you perturb them, to these very broad timescales and very broad relaxation. So this analogy, and it was really serendipity because one of the participants of our school, Ariel Amir, I suppose many of you know, did his PhD on the electron glass, and he gave a talk in our department years ago, and I looked at the graph that he presented, and his point was also how universal in physics this aging phenomenon is, because you can see it in spin glasses, you can see it in electron glass from the experimental point of view, you can see it in colloidal systems, and as well in crumpled shields. So he was pointing out that this phenomenon is universal, and the question is, can we model our cell using the same approach as is done for describing aging in physical systems? So the idea was to look at these 100,000 components, right, roughly, right? Don't take me, and do like the simplest model with analogy to the spin interacting model, and see whether we can reproduce our experimental results. So what we did was to model each of these components as a Boolean parameter. First of all, a spin, right? It's either up or down. In terms of the activity of this component, is this component active in the cell or inactive in the cell? So we have two states, and that's of course a big simplification. And now these xj's can be either plus or minus 1, okay? And the dynamics that we described for going into this high-dimensional space of all the components in the state is very simple. We assume that if J a t plus 1, and we take discrete time, Glauber dynamics, is just the sign. So it's a majority rule of so we take, for this spin, we take all the other spins that are connected to him. And here we assume at first that each component interacts, because we have perturbed the cell so much, then all these components are randomly fluctuating. We assume that each component interacts with all the other components in the cell, and with some parameter Jij. So sorry, this will be Xi, and this will be Xj. That's it, okay? And now the Jj, we take them as random parameters from a Gaussian distribution with mean 0, right? So the Jj's, and this are taken from a Gaussian distribution with mean 0 and standard deviation gamma. Okay, so that would be, of course, one of the important parameters of the model. And this is, if you look at it like that, it's really the Sherrington-Kirkpatrick model. And we could take just the Sherrington-Kirkpatrick model and go home, because actually this was shown that the Sherrington-Kirkpatrick model has this very slow relaxation and aging dynamics. But we said, okay, we've pushed a little bit too far the simplification of the cell into Boolean parameters that interact all of them all with each other. What is clearly untrue for the cell is that the Jj's are symmetric, right? In the spin model, of course, the interaction between spin I and spin J is the same as the interaction. So in the Sherrington-Kirkpatrick model, Jj equals Jji. Of course, in biology, it's not the case, right? If a component influences another component, typically the interaction back has no reason to be asymmetric and is strongly asymmetric, right? And the question is, okay, now if we take exactly these dynamics which show the aging dynamics when Jj is symmetric, what will happen for it if we just lift this symmetry and put asymmetric Jj's? Any guess? What will happen if we lift this symmetry, symmetry, and now we take the J, we randomly sample the Jj's from a random distribution without any assumption on the correlation between Jij and Jji. So what happens is that if you do that, you completely abolish aging. So now so aging in the Sherrington-Kirkpatrick model is just saying that you have a magnetic field here, right? This is a perturbation. You switch on the magnetic field. You leave it from T weight. You switch it down. You will see, here you switch the magnetic field down and up. You will see these aging dynamics. If you do it for the asymmetric case, what you see is this, and this. They relax within 2, 3 time steps. And apparently, this is something that was actually studied in the community of the spin gas model. So we could not use the same dynamics. And therefore, it meant that we needed to make some more assumptions and complicate our model a bit. Yes?
Don't we have chaotic behavior in this asymmetric model?
Yeah.
So what is the sense of relaxation then? Relaxing to what?
No, it means that, so just in here, if you look at the magnetization, right? You just look at the magnetization. You start from magnetization 0. You put the magnetic field and you put back. It's true that at the level of, it's actually like a very, very high temperature limit. So even though we do not put any random parameter, so it doesn't work. So it means that something else is going on in an asymmetric model that is is doing that. Yes?
To me, this seems that with this asymmetric J, the system is exploring more of its state space, and therefore coming back to its attractive faster. And why would that be the case compared to the symmetric one?
Okay.
I don't know this field at all, so I'm coming at this from.
Actually, I must admit I was surprised. I ran it. I thought, okay, so it's not going to be symmetric. We're going to see maybe a different behavior. Maybe this is a logarithmic decay. Maybe we'll see a parallel decay or something like that. But I must admit I was equally surprised. But it has been mapped. So I think what happens is that it just when you switch off the magnetic field after you have magnetized your spin glass, actually within one time point they do relax back. But because you have these conservation rules in the physical system, actually the fluctuation will create something that is a little bit in phase and will rebuild the magnetization up until it decays. You have this restoring force that doesn't allow the system to relax too quickly. Now, once you lift this asymmetry, actually the dynamics are much more random, and actually you see that the system just does this trajectory without any constraint, and when you take off the magnetic field, this relaxes back. This is how I view it. Yes.
When you make it allowed to be asymmetric, there's another parameter, namely the mean square deviation for the asymmetric part of J. Presumably when that goes to 0, you get back to this. There must depend on the ratio of the.
So we made it totally asymmetric, right? So that
Yeah, but you could make it.
we could make it. We can. Right. So, and actually this has been studied. People see that, yes, that's very interesting. So when you crank the parameter of asymmetry, you go from this regime where you have aging to a regime where it's more chaotic, and people that study that did not study aging, but they did study this transition to chaos in neural networks, actually.
Is that continuous transition? Discrete jump?
If it's first order or second order, I don't remember. Yeah.
Maybe it's also useful to note that, in the original shared Kirkpatrick model in this low temperature dynamics, right, you're basically trying to lower your energy, right? But the moment you make these Jij asymmetric and independent, and I is only pushed by J, and J is not pushed in the same way by I, you have a very different kind of system anyway, right?
Yes, no, I completely agree. It's not Hamiltonian dynamics, and it's very, very different. So the thing is, in order to have still to keep a simple model and restore it, we had to take from the biological picture a little bit more than just this Boolean behavior of randomly connected component. And what is clear in the biological system is that not all components are connected to each other. Actually, biology is very modular, right? And you have all these different sizes of module, which actually create a very broad distribution of timescales. So in any biological system, you have a component that can be just one component interacting with itself, to two components, to actually components that they take a big part of the network. So instead of having these randomly connected spins, what we did is actually to organize them into modules, and the way these modules are arranged is actually not important. And for simplicity, and for some analogy to metabolic network, we organize them as spins that are in loops, with still interacting, and these loops, these modules interact with each other, and these modules have a very broad distribution of sizes. And when you put that, not very surprisingly, you can reach a model that will show you the distribution of relaxation time that is very, very broad and which depends strongly on the time during which you actually do a stress to the cell. Okay? So the model actually reproduced the experimental result, and what was interesting and what is nice about the model also that it's simple enough to be completely solvable and analytically, so that it's easier to think about it. But of course, you know, I really want to, as a disclaimer, to make clear that we do not think that it is a model of a cell, okay? It is just a toy model, right? That is used in order to guide our thinking into thinking how the theory of disordered system can actually help us predict things on how bacteria or cells in general would behave under very stressful conditions. Now, what is interesting also is that, you know, you may argue, okay, you know how bacteria behave under very stressful behavior when they do these chaotic trajectories in the phase space, but maybe it's not that interesting for biology, right? Because biology is about this regulated state, right? The spore, the cell cycle. But actually, and this kind came from work that we did previously, it turns out that these bacteria that are in this state, which we call the state from which relaxation is very, very slow and very, very broad, which we call the, so as opposed to the regulated state, we call it the disrupted state. In this state, actually the cells, because they do not cycle, they enter this chaotic state. Actually, many antibiotics cannot kill these cells. And when we look back at most of the data that people have accumulated on the way bacteria become resilient to antibiotics, I'm not talking about resistance, which is a mutation. I'm talking about the fact that if you take bacteria that are perfectly identical genetically and put an antibiotic on them, you are never going to kill them completely. There are always going to be a fraction of bacteria that is going to remain. Okay, and this was something that was seen many years ago, and what we understand now is that these bacteria are actually bacteria that, instead of being in this cycle, randomly reach this disrupted state, right? And many times they are actually a memory of a starvation that was done to these bacteria in the past. They keep this memory for a very long time. This we know from the aging. And this would be the few bacteria that kept the memory of the past stress that they saw. And these are going to be the bacteria that survive. And we can really look at them under the microscope and really be convinced that the bacteria survives are the ones that are in this disrupted state. So it's not only an invention of statistical physics, it's really something that has some implication, and actually we are collaborating with doctors to understand whether our understanding of the disrupted state and of this chaotic dynamics maybe that is opening at the single cell can actually guide us to do treatment, antibiotic treatment, that you would not think about when you are looking at bacteria as if they are in this regulated state. So that's just to show that it's something beyond a game of physicists. Any questions?
So you talk about the disruptive state, presumably there are many, and there are many ways of disrupting as well.
Exactly. Okay. Yes, thank you for the question. So indeed, the toy model is so general, right? In principle, we started, initially we looked at our data on one stress, but it says that actually many stress can do it. And this is what we are mapping, and we found many stresses that before they start killing cells, because eventually from the disrupted state, you are apparently much closer to death, so eventually you will die. But before death, what we see is that many different stresses that are biologically or biochemistry annotated as completely unrelated give the same type of relaxation dynamics, not always exactly the same functional thing, but it's always non-exponential relaxation and dependence of TW. Another observation that we saw and this is quite trivial from the theoretical point of view, is that we have finite size effect, right? So what it means that if we go on increasing this TW, eventually this relaxation dynamic will actually saturate and not change, right? And clearly our system is of finite size. So what we did was to continue the experiment that we were careful not to do for too long in order not to kill the cell. We started extending the duration, and this is the third characteristic of what we see saturation at as TW exceeds about 2 days. So it as if there is for the E. coli we work with, for the conditions that we work with, these 2 days is some characteristic time scale, longest time scale of the system. And this is, and these three observations are common to these unrelated stress. So yes, now of course we would like to see how general it is also for other bacteria, of course, maybe other cells, yeast cells, mammalian cells, things like that.
I'm curious, if you increase the magnitude of your perturbation, could you just go into a different limit cycle?
So in the magnitude of the stress, so as if you increase the magnetic field intensity and not its duration, right? Yes, so this is something that we are starting to investigate because, you know, we characterize it as going from the regulated to the disrupted state. Of course, it's a caricature of what is going on, but we'd like to understand how can you make the transition between the two. And what we see, for example, for a couple of stresses that we studied, is that what is very important is both the magnitude, but mostly the time derivative of the application of the stress. So it means that if we do the same experiment and we apply the same stress, but instead of doing it abruptly, we add, so it's a chemical that we add externally, so instead of adding it, you know, we add it actually gradually. So we do it in a few steps, and then we maintain it for the same amount of time. Now what we see is that actually we have allowed the system, it's like an adiabatic switching on of the stress. So we took the bacteria from this limit cycle. Instead of going to this disrupted state, actually they go into what we call the regulated state. And from there, the relaxation is very fast, exponential, and does not depend. So then you can increase the time. The stress doesn't depend on the time anymore. Okay? And also, very similar to the spores, we see, I mean, and what we see, yes, so it's not also, but what it means is that also the bacteria are going to be much more similar to each other. They are really in this is why we think we define it as a state, because we see that the bacteria themselves, even if we started with bacteria with slightly different initial condition, they converge to this regulated state from which, per our definition, they exit exponentially and with a short time constant. Yeah, so how much time do I have left? 5 minutes. Okay, so another very important feature, of course, of living system is evolutionary perspective, right? So what is this disrupted state telling us from an evolutionary perspective? Because if it's true, and still what I'm saying you in terms of observations is solid. The rest is our interpretation, and there are people here in the audience that would maybe not agree with me. So I want to put that on the table. But what is clear is that if this picture is real, and what we have this disrupted state, which is somehow like the default state of a high-dimensional system, right? It means that okay, the biology is working hard by evolution to create these attractors, right? And these attractors in a high-dimensional space is it's very easy to break them down. All you need is to create a saddle, right, in one direction, right? And then the system will escape. So that this explains to us why very different perturbations take the cells to this disrupted state. But it means also if really we understand the different bacteria that are all genetically identical as performing a chaotic trajectory in space, from the evolutionary point of view, it means that now they can explore the space in a much wider space. And during evolution, this may be very important. This exploration may be very, very important. So this is of course, a space speculation. So one evolutionary experiment that we did in the past, and now we look back, because we are doing specific experiments now, but we've done in the past, is the following. We take bacteria, 10 to the 9 bacteria, and we stress them, but in a gradual way, so that they enter the regulated state in our framework. Okay, so they stop their growth, but in a regulated manner. And then we dilute them in the morning into antibiotics. Antibiotics that completely kill growing state, right? So these antibiotics, if you are in this limit cycle, you die. But if you are not, if you are arrested, either in the regulated state or in the disrupted state, you die. But we take them from this regulation, and we actually let them relax because we give them energy and food here and mass, right? So they all wake up. I mean, they all relax fast because this is a regulated state. So no aging here, and therefore they die a lot, right? And they die at the beginning of the experiment, they die by 5 orders of magnitude, so the survival is 10 to the minus 5. What we think is that the ones that survive are stochastically in this disrupted state, but we haven't shown it, okay? We leave it for some time, and then we let the bacterial population, we wash the antibiotic and we let them grow again, and again go into this growth arrest, okay? And we do it every day. And after a week, even less, a few days, what we see is that the population, instead of having this very low survival rate, now the survival rate is 10 to the minus 1, even less than that, okay? So it's even more than that. So they are hardly killed by the antibiotics now, okay? And they are hardly killed by the antibiotics without having acquired resistance. So they are not able to grow in these antibiotics. And the probability to be resistant in this antibiotic is very low. So we knew that resistance will not evolve. So what has evolved? It did acquire mutation, a lot of mutation. I mean, each bacterium acquired different mutation. And these mutations, actually, the main result of the mutation, apart from allowing them to survive, is that now if we take the bacteria, so now if we compare the ancestral before we evolve it, so without mutations, and we grow it and we put it and we let it starve, it goes into this regulated growth arrested state. Now if we look at the distribution of time to re-enter the cell, so the single cell distribution of time, what we knew is that it's something that is quite narrow. They are in the regulated state. It's an exponential process, and the typical time scale is about 40 minutes here, okay? So it's narrow and 40 minutes. Now if we look, and this is still 10 to the minus, as a survivor of 10 to the minus 5 when we put it into antibiotics of a certain duration. Now what happened in the evolved strain is that now this is actually showing the relaxation dynamic of an aging cell, okay? So it has this very broad distribution, and this broad distribution actually allows it to survive the antibiotics, because if when if when we expose the bacteria to energy and mass and antibiotics, right, they don't feel the antibiotics, they don't respond to it, so they start growing, they're dead. This is why we have this very low survival in the wild type. Now, these bacteria now that have these aging dynamics, right, when we expose them to food, they take much longer to relax. Let's say the mean here, for example, is can be shifted from 40 minutes to 4 hours, right? And now if you put an antibiotic treatment of 3 hours, right, all the bacteria that were actually that relaxed after 3 hours will actually survive, and therefore the survival is much, much higher, right? So, and at the time we varied this time where we put the antibiotics. So if we put 3 hours of antibiotic, we get this distribution. If we put 5 hours of antibiotic, we get a longer distribution. If we put 8 hours on antibiotic, a longer distribution. So they really adapt the distribution to the duration of the antibiotic stress that they've seen before. But, and at the time, we saw it as a nice example of evolutionary adaptation. But now, what we understand, we think, is that actually what happened is that just one single mutation, these bacteria, all these clones, when we sequenced them, they had this capability of having this very wide distribution of relaxation time just by having one mutation. All we understand is that now these mutations actually take the cells from a regulated state to the disrupted state. So this starvation that was slow enough to maintain the wild type, I mean the ancestor strain, into a regulated state, now is actually disrupting for this mutant, and therefore this disruption actually paradoxically enables them to be slower and therefore to survive antibiotic treatment. So not surprisingly, it explains to us also why we see all kind of weird mutations that are not really related, because as I said, disrupting the regulated state, which requires, you know, all these dimensions to be perfectly controlled, you can now perturb and disrupt the system in many ways. So many different mutations targeting one of these axes or many of these axes will make the bacteria evolve into the disrupted state. Oh, thank you. Yes?
Just based on the picture you just drew for us, what if I give my antibiotic treatment for the same amount of time, just wait longer before the next one? Is the survival ratio then lower? Based on the fact you said this is not really an adaptation to the antibiotic, it's just that they're aging back from this disrupted state. If I let it age longer, to Europe and survive.
Exactly. No, yes, of course. If we take bacterium that have evolved under these daily cycles of 3 hours of daily cycle to antibiotics, and we put it for a day into antibiotics, it will not survive much better than the wild type.
If we wait longer and then still again put it into the 3 hour back,
yeah.
Would they also be?
Yeah. Yes.
Okay,
you need.
This is reverting back to their original status, and okay, so that shows.
Right. As soon as they start growing again, they are killed by the antibiotics the same way as the ancestral strain. Yes.
And maybe another question, and this is maybe my lack of knowledge field, but when you're drawing a Boolean thin glass model, but not really a glass because there's no energy, so it's not symmetric anymore. What is, even in the absence of the field, magnetic field, what's the state that's the base state, ground state of it? Is it a limit cycle because you're drawing a parallel to a bacteria?
No, it's really a chaotic state, and I must admit that we don't have much to say about it. Yeah, I mean the magnetization is 0, right, because of the symmetry, right, of the problem. But
you said there's no aging, so you can find whatever that state is pretty quickly. It's just, I mean, I don't really see how this, other than the fact that you get aging when you integrate it modular. Why does this reflect your bacterial dynamics? Like, what's the?
Okay, so when we make it modular, we retrieve the aging dynamics, and we can retrieve also this saturation, which So far, the model described our bacteria, and this is a very, you know, a caricature of a cell, of course. But now what we want to probe, how far can we go with this model, which is clearly wrong. So everyone who said that the model is not describing a cell, I agree. Right? The question is it useful? Can it really help us? Now, we have, for example, now some prediction from these dynamics at the single spin level of the model. We have prediction for, if we look now, because we can label these dimensions, some of the dimensions in cell, we can look at the dynamics. So there are clear prediction of the correlation between these spins, these degrees of freedom, that should also show these aging dynamics. So this is what we are going to probe now, right?
Yeah, so I guess I also have two questions, but first let's clap. I think this is super stimulating and appropriate. Two questions. One about biology, and but the first one about symmetric and non-symmetric models. So, right, this is spin glass in a landscape with many minima, relaxed. So we can think of these as different states of the cell in some Waddingtonian sense. But you seem to make a point that no, it's not a landscape, it's dynamics, but with perhaps chaotic, but not ergodic.
Not ergodic. No, not ergodic.
Is that what you're saying? That this model with connected cycles
okay.
is actually chaotic but not ergodic?
Yeah. Okay. So.
Is it important, or are we just recovering lack of ergodicity in that partially symmetric or sparse model going back to a spin glass landscape?
Okay.
Is dynamics important, and perhaps the question is there some way of finding out in actual microbes whether it's Waddingtonian or who invented chaos? Or chaotic?
Oh, Lawrence. So, okay, so it's a fantastic question, right? And I forgot to say, but it's a great point to take it. All the theoretical work was done in collaboration with Oded Agam, who is a condensed matter physicist and statistical physicist in our department. And these are exactly the questions that we would like to investigate. And actually, I think what we run into is that there is very little solid ground for in theory for these kinds of systems and dynamics, right? There are very little that we know that we can say about these systems, and this is definitely something that we'd like to investigate. If we don't have ergodicity, we are in trouble, right? But something that we can investigate at the experimental level is really take for example the regulated state and perturb it and measure the stability of this state and do the same measurement for the disrupted state and see what is a kind of response dynamics and see how different it is and try to see parameters that will at least constrain the next model that we'll need to develop for that. But yes, we just at the beginning.
And the second question was biology. So disrupted state. So do you know anything about physiology of the disrupted state in the sense, are these cells just expressing transcribing random genes that they wouldn't do in a regulated state, but that you think is accessible to?
Yes, so for example, something that we started doing, but it's really very preliminary, is that we take one of these axes, right? One of the major ones, like the production of ribosomes. So it's and we see when it becomes active. So in the regulated state, if you look at this, you see nothing. It's a regulated growth arrested state. No ribosomes are produced, and if you look at many different cells, they're all off. In the disrupted state, what we see, but it's really preliminary, is that suddenly you would see one cell going up and down, and then another one going up and down, And this is, you know, this is the first indication that there is, so it's clearly a dysregulated state. I mean, this is what, how we interpret it, but of course we'll need more than one.
What you said already points to dynamics.
Yes, exactly. Yeah.
Just a naive question. So if you take, let's say you take one of these highly reduced bacteria, like what Greg Venter has been doing.
Right.
And try the same game, what happens?
Ooh, that's a good question.
They kicked out so much.
So much, right. And you took them so far away from their evolutionary.
Reduce the dimension roughly.
Yeah, it reduced the dimension, yeah.
So what would happen? Sounds like.
Still, I think they have 100 genes, right? How many of these mycoplasma have? I think 100. Have 100 genes now.
They still have a very high dimensional, but is there regulated state for them or not?
That's a very nice question. I don't know. What is very different in these bacteria is that in our bacteria we have this very high dimensional space, and if you add the access of external inputs, for example energy and mass, they are, you know, all of one. So they are basically on only a few access compared to the 100,000. For the bacteria you're talking about, they can live only inside one of our cells, which has back these hundreds of thousands of components. So it's a bit different, right? So now their dimensional space is embedded into a much higher space, so it's difficult to. So maybe there it's a different limit. Yes. Thanks.
Any more questions? Okay. Well, thank you very much.